Welcome back to the course Fundamentals of General Immunology. We'll start today a new topic that is related to mucosal immune response. So we'll name that module as Mucosal Immunology. And in Mucosal Immunology, we'll try to look into the parts of the body that's heavily supplied with the mucous membrane or the mucus secreting cells. These are generally the areas of gut, respiratory epithelium, genital tract, and to some extent the parts where the surfaces are mostly moist. So let's move on and see the details of mucosal immunology. So first we'll try to learn about regional immune system. When it comes to regional immune system, they are basically localized immune responses to some very specific organs. For example, the gut epithelial wall, the skin layer, the lungs, and the genitourinary tract, or the specific part of the genital as well as the urinary tract. Generally, these are exposed. So basically, gut is getting the food particles from the mouth or oral cavity. The skin surfaces are always exposed to the harsh conditions, where temperature, moisture, and say some other kind of external stimuli. Lungs are also kind of getting all the time the air that's coming out from the outside. So these are also kind of getting a constant environmental pressure. So managing the commensal microbes without causing excessive damage is the utmost function of these areas, and the immune system can take care of all this very critically. Now, they generally differ from the other immune system by generally operating independently to provide protection against food specific allergies and also airborne specific pathogens. So when it comes to the food borne allergies, then we talk about gut, intestine, and the specific things which are coming through the oral cavity. When we are talking mostly about the airborne pathogens, then generally lung, upper respiratory tract, nostril, and these areas are basically involved in that. So this is what is meaning of the regional immune system, because they are localized. They are particularly concentrated towards a particular site, and they are generally getting an environmental pressure most of the time in various forms, either in the form of a food or in the form of an airborne pathogen or allergens, or also there could be some other specific thing that is leading to some kind of immune intervention in the human body. So these are very specific and specialized mucosal surfaces. When it comes to mucosal surfaces, defining mucosal surface is, in a layman language, one can understand that these areas are mostly moist, so they have high concentration of the water in that area. So epithelial membrane lining the internal body cavities such as the gut wall and the complete alimentary canal, the nostril, upper respiratory tract, genitourinary tract, and the inner lining of the skin area. So these are basically the internal membrane lining of the body cavities, mostly the body cavities. And as such, we discussed that this includes the respiratory, gastrointestinal, and urogenital tracts. These surfaces are critically barrier and the entry points for many of the things, and therefore we need to have a very robust immune system in order to protect the pathogens which are entering inside the body system through these openings, because essentially these are exposed to the openings. So the nostril is giving a direct entry to the pathogens to the lung, which is an internal organ, and again the oral cavity gives the entry to the alimentary canal, urogenital tract, and other things, because that's kind of an entry point. And they house the majority of the body immune system or body immune cells, but there are variation. We see what are the different variation that's kind of different than the systemic immune response which we have discussed till now. So that is what is different in mucosal immunology. We'll discuss about tolerance as such, because the food particles which we are taking is not raising an immune response, although they are antigens, and therefore there could be some sort of tolerance against these food particles in the human body, and therefore we are not mounting an immune response. Otherwise, once we are taking any food, since they are antigen or these are coming from the outside, we should immediately vomit it out. And that's not happening, because our body immune system, the regional immune system of the gut, is taking care of all those food specific particles as a kind of not considering them as a non-self, and therefore they are not raising an immune response. So this is different as compared to other systemic immune response. Now let's see the mucosal cell types. So we talk about the mucosal surface rich in moist and say constantly under pressure with multiple external things. So we'll look into the mucosal cells, how these are different than the other cell types. So generally they are called primarily the stratified squamous epithelium. So stratified means layered. So we have 1 layer, 2 layer and 3 layers. So if you are getting this kind of pattern, we call it stratified. That means there is a layered. Squamous is just a type of epithelial cells. We define the types of epithelial cells in different types of the cells, which we'll see in the next slide. These are generally the area where you always see constant friction to happen, such as mouth and esophagus, because mouth is taking the food particle, chewing it with the help of saliva, and then constantly in frictional environment as compared to any other type. Esophagus is also a food pipe. So basically the food is passed through the esophagus and reaching to the stomach, and without the friction to happen, that's not possible. And therefore this is also an area where you can find a lot of cells which could be a stratified squamous epithelium. So generally you find those areas where you have most of the secretions coming out. Those all cells are mostly squamous in nature. Very flat cell, we'll see in a while how these look in anatomical way. Then comes to the columnar epithelium. As name itself indicates, these are very tall like cells, the column like cells, and the nucleus may be in the bottom most of the time. So they can mostly present in the alimentary canal, because they might have the brush border, say they may be ciliated, and therefore the cilia can help in the movement of the cells and basically allowing the passage of the food from forward to a backward direction. So basically if it is a peristaltic movement and with the gut food is passes through this direction, these cilia can help to propel the food in the forward direction. So these may be ciliated as well. So this columnar epithelium are mostly present in the lining of the stomach and the small intestine and large intestine as well. These are mostly responsible for absorption as well as secretion. There are many other cell types which are present. We'll gradually learn about that also. Then we have something called pseudo-stratified columnar epithelium. We'll see in a while. This type of epithelium you generally find in the respiratory tract, because they give a feeling of a stratified, but they are not stratified. Say for example, if we have this kind of a structure and then you have this kind of a structure, although these are a single cell, but they are in the same size of the two layered cell, and therefore this gives a feeling of the multiple layer, but these are pseudo-stratified. Pseudo-stratified means false layered. Pseudo means false, stratified means layer, columnar epithelium. So this could be a different columnar epithelium, and that's present mostly in the respiratory tract. Then we could have a transitional epithelium. So transitional epithelium, and these epithelium are mostly present in the urinary bladder, because these are, as name itself indicates, they are transitional. That means they can transition from one type of cell to the other type. They are flexible because of the nature of the urinary bladder. So once the urinary bladder is filled, their volumes are very large, and therefore we see the cellular morphology to be in a way so as to accommodate more and more fluid into that organ. When it is empty, then you need to reflex that organ so that the cell morphology is coming back to their original size. So therefore you see flexibility of the cellular type based on the volume of the water inside the urinary bladder, and therefore these cells are called transitional epithelium. So you see those kind of cells in the urinary bladder, and there are other cell types as well. These surfaces are moistened by the high concentration of the mucus, because you have a goblet type of cells which can secrete mucus in the alimentary canal. We'll see in a while. And the mucus is a huge barrier to anything coming and touching to the surface of the alimentary canal. Say if you have a columnar epithelium, there is a thick layer of the mucus on the top of it. So if there is a layer, and if the bacteria or any kind of pathogen is coming into sight, they are not able to touch here on the surface because of the volume of the mucus, and therefore they can propel forwardly with the help of the mucus, and therefore we are protected by any such of infection by those bacteria due to the mucus surface on the lining of the alimentary canal. So these are some of the cell type. Let's see and understand how these look like in morphological way. So the first one is squamous epithelium. There are two types of the squamous epithelium. One is called simple, which is simple layered, right? So you can see that this is a very flat cell, and the nucleus is just bulge out. It's such a flat structure that even the presence of nucleus can give a bulging appearance to the cell. It's like an omelette sort of thing. So the cell membrane is bulging because of the nucleus. It's a single layer, so you call it simple squamous epithelium. These are generally present in the area where you gradually see huge amount of secretion coming out, such as eye. So you see that tears are coming out all the time from the eyes if something is coming in front of our eyes because of the sensitization. So the lacrimal gland will secrete immediately those fluid into the area. So these are always moist. These are very sensitive cell types. Again, there could be other cellular areas in the buccal cavity. You have lining of the buccal cavity that's also having a squamous epithelium. So squamous epithelium is mostly located in an area where there is a requirement of secretions. The same we discussed about, there could be in a stratified layer. Multiple layer of the squamous epithelium can make it as a stratified, and these are present mostly on the area where you see friction to happen, such as tongue, and also in the buccal cavity other area along with the esophagus. So these are one type of cell which is typically involved in secretion. The second is columnar. As we stand, that columnar could be a simple columnar, a single layer, and then you have a cilia or the smaller ciliated form, we call it microvilli, and these are giving an access of movement to the fluid present on the top of it. So these are present in the lining of alimentary canal. Then we have a stratified columnar. Stratified means again you have a layer of the cell. So you have one layer, and then on the top of that thing you have another stratified layer of the columnar epithelium. They can be required on an area where you have a thickness of the columnar epithelium, and therefore these are giving a strength to epithelial area. So this is second one, columnar. Mostly it's present in alimentary canal. Then we have third type called cuboidal. It's kind of a similar like columnar, but the cell size is smaller. So therefore they look like a cube, and therefore they are called cuboidal epithelium. Simple cuboidal epithelium, and then you also have a stratified cuboidal epithelium. Cuboidal epithelium are mostly present in glands. So glands are the area where you see hormones and other things which are stored in order to do function in a proper metabolic way in a human being. So these are mostly present in the glands, such as the individual glands which can secrete some, such as salivary gland for example. They can secrete the saliva on the top of the tongue in order to moisten the food particle, and also containing a lot of enzymes such as amylase to digest carbohydrates and stuff like that. So these are mostly a lining of the glands is built by this cuboidal epithelium. Again, if you need more strength, you require a stratified layer, more layered structure. So this is stratified is just a term which is given anatomically to the organ or the cell type where you require more rigidity, more strength is required, then you find out stratified as a solution there. Then we also discuss about transitional epithelium, that's present in the urinary bladder, and you can see multiple different types of cells. Some looks like cuboidal, some looks like a squamous, some look like a columnar, right? So these can transition. The cell can transition from one form of the cells to the other form in order to do functionality, proper functionality of that particular organ. So this is transitional epithelium. And then we looked into stratified columnar epithelium. The term added here is pseudo. Pseudo means false. Now you carefully look into this picture where you can see that they are different cell type and their nucleus is located in a different way. They give an appearance that you have a single layer or columnar epithelium, right? But they are not columnar in nature, but the structure look like a pseudo structure, because the nucleus are all located in different directions. So they are not generalized structure, and therefore they are called as pseudostratified. They look like a layered because of the presence of nucleus. So one can see that, one can always say that there are two layers, because that is one bottom and the one is on the top. However, the cells are same. So this is what is stratified, particularly pseudo, false stratified columnar epithelium. Now we will try to learn a little bit more detail about the environmental factors and the distribution of the microbiota in the gut. So we generally know that there are huge microbial load in the alimentary canal because of the useful character of the useful bacteria of the alimentary canal, because they are required in order to do some processing of the food particles, secretion of some enzymes, vitamins, and very essential component that helps in digestion of the food particles. So the first one is stomach. Now stomach is basically acidic in nature. You know the pH of the stomach is roughly 2 to 4. And therefore you find the bacteria which are available or present in the stomach could resist the pH as well. So some of the bacteria such as lactobacilli can be found in that particular area. Not that in a very high concentration, but to some extent you can find lactobacilli there. Then we have a duodenal area, and duodenal area also, and jejunum is kind of an part of the small intestine. So the small intestine part is divided into duodenum, jejunum, and ileum, and then finally you get a large intestine, which is cecum, colon, and rectum. So this area is loaded with different concentration of bacterial population. So we start that in the stomach, because of the higher pH, we get a very less concentration of bacteria. Little higher in duodenum, jejunum, and you go slowly down, you see more and more concentration of the bacteria. And the highest concentration you get in the digested food area, where you can always see higher concentration of bacterial load in the lower or terminal part of the large intestine. And these are the one which allow the unwanted food to pass through the stool out from the body. And therefore people looked carefully into what kind of population of bacteria there might be in these compartments, and some of the bacteria such as lactobacilli, which is a very useful bacteria, streptococci, and these are actually resident of the alimentary canal. Apart from that, we have Clostridia and some of the typical bacterial population which is listed here. We will not go and discuss everything here, but just to add a point here, that we know that these bacteria can degrade the complex polysaccharide and other undigested fiber into small chain fatty acid to give energy, and these small chain fatty acid are very important component for T regulatory cell population, because if we are down regulating the small chain fatty acid, so small chain fatty acid concentration can maintain the T regulatory cell, which we will see little later, that once we have a lower concentration, we get a lower concentration of T regulatory cell, and therefore we won't able to raise a immune suppressive or immune tolerant environment in the gut. So therefore the small chain fatty acid concentration is important. These are coming because we have this bacterial population that can digest the fiber to make this small chain fatty acid. And therefore, there are sentences written here that these bacteria use complex polysaccharides. Complex polysaccharide means many kind of carbohydrate moieties and other component of mucus and undigested fibers as energy sources. So they can use all the important content of the fiber and some component of carbohydrate as an energy source, and they can convert these into very essential components such as biotin, short chain fatty acids and vitamin K. So these are essential. Any deviation, any malfunctioning of these bacterial population in the gut can lead to malabsorption and less production of these essential components such as biotin and SCFA, leading to more complicated conditions, and also the particular thing towards the immune intervention as well. When we talk about T regulatory cell, oral tolerance, then we'll understand the role of short chain fatty acid there itself. So these are some of the things which are important. Let's move on and see one by one that what is there in the small intestine and the large intestine. So this is actually a area of jejunum. We call this, so we can take an image of the jejunum by putting a small pipe containing the camera. So that's called endoscopy. So once this is penetrated inside an area, we can see that there could be a cell type which contains a very high amount of breast border epithelium. We can see that there are huge amount of ciliated epithelium. So these are called breast border, and they can be very rich in this goblet cell. You can see these small cells which can secrete large amount of mucus. So the mucus secretion depends upon the goblet cell population. If this is the goblet cell population is less, we see less mucus secretion in that particular area, and therefore the cellular health will also be compromised. So this is actually hematoxylin and eosin staining. So this is also called as, in short, H&E staining. That's generally done when you take a cross section of the, suppose we have this tissue, suppose this is alimentary canal or any organ, for liver also for example, you take a section, and once you take a section you can put it into a slide and you can add this hematoxylin and eosin stain, and after staining you can see those specific architecture of the cellular system into microscope. So this is just a method to visualize the cellular mass or cellular structure under the microscope. So this gives an indication that the health of the cells are intact. So this is just an appearance of the jejunum, which is a part of a small intestine, and looking into the breast border and the goblet. Of course, there could be a lot of other cell type which we'll discuss in the next slide. Then we could understand that this is not only one cell type, there are multiple cell type, but this is just an H&E staining that gives an indication the rough architecture of the small intestine. If we move into the mucosal surface of the small intestine in a broad way, then we'll see that you have many things there. So of course there will be a commensal bacteria which could be present in the alimentary canal. These are secretory immunoglobulins which are present and secreted in order to bind to something which is coming into the circulation. We also have an intraepithelial lymphocyte which are kind of an immune cells which can secrete immunoglobulins. This is actually a mucosal layer, and then we have a small muscularis mucosa, then we have submucosa, and finally we'll have a muscle layer in the bottom, and then we'll have a blood vessel as well below this. So these are the three layers. We discussed that the goblet cells are the one which can secrete the mucus in the area, allow the fluidity to happen. There could be cells which is called stem cells. These are the progenitor cells that can produce multiple different cell types. There are cells which are called Paneth cell also, which can secrete lot of antimicrobial peptides. So these are actually the small stretches of amino acid stretch which can help to fight against the pathogen and also doing some physiological function. Apart from that, we'll have macrophages, dendritic cell, the B cell which can secrete immunoglobulin A, T cell, and some other immune cells. Apart from that, we'll also have an alimentary canal cell called enterocytes, which are basically a cells which contains the microvilli and that can help in absorption of the nutrient from the cell, and also these intestinal epithelial cells which are getting apoptosed and getting replaced by these kind of stem cells which are present in the alimentary canal. So this is a complex area. Apart from that, you also get some of the enteroendocrine cells which can secrete multiple hormones which can control the, say, movement of the gut wall. So these are very complex architecture of the small intestine that allow the absorption of the food particle, and also we focus here on the structure of the immune system here. Apart from that, we'll discuss later on that we'll have a T regulatory cell also present here, which can secrete important cytokine that can lead to immunosuppression, and therefore we learn a technique called oral tolerance, which is actually ability of a human being to tolerate against the food that's entering through the alimentary canal. All right, we'll move on and see to the large intestinal wall. So this is actually the one of the typical part of the large intestine called colon, and you can see the flat surface of the colon. That means the breast borders are not there, so the roughness is not there. We can see a lot of absorption of the water in this particular area because unwanted water will be taken out from these digested food so that the water can be retained back to the body system. And we can see that the goblet population is more, so we can have more mucus secretion here, and then there is no or very less amount of breast border in that. So this is roughly a kind of an architecture of a large intestine. And when we see grossly, you see that there could be a lot of goblet cell which can produce a mucus, and you see a mucus lining. Apart from that, we have the apoptotic cell and some of the resident macrophages and antigen presenting cells, along with some B cell and T cell population as well. And antimicrobial concentration will be a little less in this particular area because these are rich in commensal and rich in some of the bacteria which are present in the large intestine. Now this is roughly architect of the large intestinal and the small intestine, which are involved in the mucus secretion and the gut wall as such. So we are trying to look into the mucosal immunology, and to understand mucosal immunology we need to understand what are the things which are involved in making these surfaces intact. So as such, when we talk about the mucosal surfaces as an immune barrier, there are cells which are present there, and they are collectively called as mucosa associated lymphoid tissues. They are called MALT, M-A-L-T, and these MALTs are characterized into different parts based on their locality, such as nasal associated lymphoid tissue. That means they are present in nasal area. Gut associated lymphoid tissue, G-A-L-T, GALT. So NALT is in the nasal cavity. Then you have a gut associated lymphoid tissue which are present in the gut wall. Then we have a bronchus associated lymphoid tissue which are present in the bronchi or in the respiratory tract. Then we have lymphoid tissue associated with the genitourinary tract. So there could be a separate lymphoid system which can present in the genital tract and the urinary tract as well. So these are some of the types of the mucosal barrier or mucosal tissue which allows secretion of different cytokines or different mucus layer, and altogether different immunology in different areas where they are present. Now finally we look into the roughly how it works. So suppose we have this antigen present in the gut wall or gut area. These antigens are processed through these very specialized cells which are called microfold cell. So they are basically the antigen presenting cell. They can pass on this antigen presenting cell to the lower area which is called subepithelial dome, and in subepithelial dome we see the antigen presenting cells to be processing those antigens and then activating the B cell or the T cell immune response at that particular site. And then we also see that we will have this follicle associated epithelium on the top, and then we might be seeing so this is a structure of the gut associated lymphoid tissue, and we could have a germinal center as well. So once there is a activation of the immune response against these antigen or bacteria that enters into the body cavity through the gut, we see these immune response are drained through the lymph to the site, and then we see that they can be disseminated and start the effector function by producing a very specific secretory immunoglobulin, which is IgA in nature. So essentially, once we sensitize, so once the follicular associated epithelium is sensitized from this antigen, then these antigens are processed through the M-fold cell, microfold cell, and processed by the antigen presenting cell, tuning the T and B cell to produce more and more cytokine and immune regulatory cells along with the antibody. These are drained through the lymph node and then do the function at the distant site by producing amount of IgA in order to neutralize those bacterial population. So we can see that we could have a macrophages, plasma cell, secretory immunoglobulin. Of course, we are trying to see a dimeric form of an IgA molecule, and this can do an effector function in the alimentary canal in a very, very precise way. So this is very distinct immune response as compared to the systemic other immune responses where we will talk about IgG and IgM immune response, but here we will talk about mostly secretory immunoglobulin such as IgA, that too in the dimeric form. So this is all about the rough structure of the mucosal surface, what are the cells involved there, how it is different than the other body types, and how these are grossly expressing the immune specific regulatory molecules upon encountering an antigen. So we'll talk more about this structure closely and try to learn more in our next class. Thank you.